Basically, we use two animal models for our study research. One is a reptile, oviparous lizard, Calotis versicolor, and Swiss albino mouse. Current research in our department is molecular mechanism of sex determination and differentiation in the oviparous lizard reptile, which is mainly concentrated on temperature dependent sex determination, and using a mice model, biochemical and pharmacological impact of anabolic androgenic steroids in female mice and endocrine, and this aspect we have recently started. Now coming to the sex determination, which is a very interesting aspect from we are using a reptile model since many years. And as you know, first I'll give a brief introduction about the sex determination mechanism. In vertebrates, sex is determined essentially by two means: genetic factors located on the sex chromosomes, and which is referred to as genotypic sex determination, and epigenetic factors, for example temperature, mainly most of the time, and which is called usually environmental sex determination. Most predominantly it is temperature dependent sex determination. In GSD, as you know, the sex of the offspring is determined at fertilization and obeys two main system. This is very basic, which follows male heterogametry in case of as in mammals, and female heterogametry in birds, amphibians, reptiles and fish. And in ESD, it is determined permanently after the fertilization. That is the main difference. It is at fertilization, sex is determined at fertilization. It is at after the fertilization by the influence of environmental factors, and mainly the temperature, which is referred to as temperature dependent sex determination. But we are interested in what are the molecular players in translating temperature into an endocrine signal leading to the steroid production. In the process is still in reptiles, it is still unknown. See, it is many oviparous reptiles, including all crocodiles, most turtles, some lizards, and few even viviparous gametes exhibit temperature dependent sex determination, wherein the temperature and the hormonal milieu, mainly particularly the estrogen, determines the gonadal sex of the embryo during a critical period. This is important. Only these are acting. Temperature and the steroid hormone milieu, especially estrogen, acts during a critical phase of the development, and this is known as temperature sensitive period. It acts for a few days, and after that the temperature is will not be effective. For example, the temperature mainly acts on the what I referred as estrogen, where actually the aromatase plays an important role. Testosterone gets converted into estradiol with the help of the aromatase. Okay, so it acts actually like this. So before the TSP, temperature sensitive period, the embryo will be having undifferentiated gonad. It is not yet differentiated in either into a testis or a ovary. It is going to differentiate. But during the TSP, what happens exactly? High temperature leads to a high aromatase activity, which is leading to the formation of females or the ovary formation. And during the this period only, if there is a temperature is low, then it leads to low aromatase activity, down regulation of the aromatase activity leading to the testes. So this is high aromatase. After this TSP period, there is no high aromatase activity or temperature effect will not be there. So what is the embryo experience during this period? Hormonal milieu or the temperature determine the sex of the embryos. So work from our laboratory. We work mainly on the Calotis versicolor, and this is of excellent model to study the endocrine and molecular cascade since it lacks the heteromorphic sex chromosomes. Then we are interested in what might be the factors regulating the sex of this lizard. Then we tested the temperature when it lacks the heteromorphic sex chromosomes. Then we generally people go for the other ESD factors. So during that period we have tested the temperature sensitivity of the lizard embryos by incubating at a broader range of temperature, by incubating the eggs at a broad range of temperature, and the resulting deviation of the sex ratio, Mendelian sex ratio, reveals that it is having a TSD. So in that process we got very interesting results, whereas two temperature produces the 100% male producing temperature, and these two temperature produce the 100% female producing temperature. Then we referred it as male producing temperature and female produced temperature because they produce the 100% of the sex ratios. And during this meantime, we got the very interesting results where we are the first, our lab is the first to give the FMF pattern of temperature dependent sex determination. Actually, in TSD, there are 3 patterns existed. So, one first case where males are produced at high temperature, 100% males are produced at high temperature. This is case 1, MF pattern. In the second, the reverse is true, where 100% females are produced at a high temperature, FM pattern. In the third case, FMF pattern, where females are produced at too extreme temperature and at intermediate temperature males are produced. Ours is entirely different, where 2 females are produced at too extreme temperature, low as well as high, and males are produced exactly at low as well as high, which is we named it as FMF pattern. So, we tested with the help of the histological figures. This is the TS of the ovary, where it is cortex and medulla is there, and even with the help of the accessory reproductive structures where Mullerian duct and Wolffian duct. So, this is in case of generally in case during the development, both embryos will be having both Mullerian duct and the Wolffian duct. With the development, in case of males, Mullerian duct regresses with the help of the Sertoli cells, which secretes Mullerian inhibiting substance, anti-Mullerian hormone. By that, Mullerian duct regresses and the Wolffian duct develops. Here it is Wolffian duct. This is the case of the testes, accessory reproductive structures, this is kidney tubules, Wolffian duct is developed in case of males and the Mullerian duct is regressed. In case of females, Mullerian duct will be developed because of there is no secretion of Mullerian inhibiting substance by the Sertoli cells. Lack of Sertoli cells in case of females retains the Mullerian duct, whereas in case of males, Sertoli cells secretes the Mullerian inhibiting substance where this Mullerian duct undergoes regression. See this exactly at hatching, where cortex is more developed, proliferates in case of females, and medulla in case of males. And here Wolffian duct is developed, Mullerian duct is developed in case of females, and here ruminant RMD, ruminant Mullerian duct, Wolffian duct is developed. In case of females, Wolffian duct will be, Mullerian duct develops. And see, and when we got this interesting pattern, FMF pattern, but earlier reports from our laboratory states that the morphological differentiation of the gonad precedes the onset of steroidogenesis. See, morphological difference, before the morphological differentiation of gonad, there was no steroids. Then we tested what is the source of steroids. If there is no gonad is not secreting the steroids, then what is the source of steroids? Is there any extragonadal source? Yes, we tested for 3. Maternal yolk steroids, the adrenal kidney complex. Why adrenal kidney complex? Because gonad is attached to the adrenal kidney during the development. So, is there any source of extra paracrine effect of this adrenal kidney for the onset of steroidogenesis in case of gonads? And then we got very interesting results in the brain neurosteroids. So we this is the first stage of a reptilian embryo where we tested the yolk. Then this result showed that there is no correlation between the sex ratio and the yolk steroids. So we came to the conclusion that yolk steroids are not the decisive factors in sex determination mechanism. However, maternally derived steroids may act as initial metabolic sources. And then we went for the endogenous steroids. If there is no maternal steroid, exogenous steroids, then we tested the endogenous steroids, mainly from the steroids of adrenal kidney gonadal complex. Then we got the very interesting results where this graph reveals that testosterone level is high at male producing temperature and estradiol level is more at female producing temperature. And so it is inferred that in conclusion our experiment said there is an apparent surge in the endogenous E2 level coincides with the first indication of the gonadal sex differentiation in this lizard. So it is inferred that at this temperature aromatase may be upregulated, and increase in E2 concentration and decrease in testosterone level during TSP suggests that there may be an interplay between the steroids and the temperature. Furthermore, when their steroids are there, then what is the source of, which one is the source of steroids? When there is no gonad is secreting steroids, then when estrogen is very playing an important role, the source of estrogen in the reptile embryo, however, it is still controversial. So in this context, we examine the expression analysis of estrogen receptors, ER alpha, aromatase in the embryonic adrenal kidney gonadal complex at FPT, means female producing temperature, and the male producing temperature. And here we got very interesting ER alpha, expression of the ER alpha in the kidney tubules as well as in the adrenal. So this is the expression of ER alpha in the medullary region of the gonad and the cortex portion. See, during the development, adrenal will be attached to the gonad, where we got the ER alpha expression in the adrenal cortical cells, not in the medullary cells. Adrenal cortical cells and the medulla of the gonad. Here the expansion of the same figure, where medulla of the gonad and cortex portion, cytoplasmic expression, not the nuclear expression. So the estrogen may be, it is present in receptors, may present in the cytoplasm, where see here in the kidney tubules, here the cytoplasmic expression of the ER alpha, there is no nuclear expression. And it is the TS of the testis showing the ER expression in the seminiferous cords. Usually during the development, it is we refer it as seminiferous cords. Tubules are not yet formed, and this is the ER expression at the where tubules are formed. Seminiferous tubules are formed at nearing to the. See the ER expression is the enlarged view of the previous photograph, where adrenal will be showing the ER expression in the adrenal cortical cells, in the medullary cells of the gonad, and it is started separating. The gonad is started separating from the adrenal. So we thought it is, but still attached to the adrenal. So during development, adrenal steroids synthesized, maybe adrenals may be synthesizing steroids, which helps in the, which gives a paracrine effect on the gonad, so that this extragonadal source of steroids produced by the adrenal and the kidney may be helping in the process of gonadal sex determination and differentiation. Here again the same figure, which is enlarged view. This is a Mullerian duct. So by the later development stage, even the Mullerian duct will be showing the estrogens. It is showing its signal to the estrogen signaling, showing it, and this is the main. And here during the later stage, where the ER alpha expression is found in the kidney tubules, and the nuclear expression shows that these are also synthesizing estrogen. So immunolocalization. The same thing I told, the ER alpha in the medullary region of the gonad showing weak cytoplasmic expression, and then later it shifted to the nuclear expression. Then we went for the Western blotting analysis of ER alpha, where it shows the ER antibody recognized two protein bands of 55 kDa and 45 kDa, revealing the presence of two isoforms of ER alpha, which is the first report from our laboratory during the development. So Western blot analysis revealed two protein bands with apparent molecular weight of 55 kDa and 45 kDa in a total protein extracts of adrenal kidney gonadal complex, suggests the occurrence of isoforms of ER alpha. And the increase in expression, see the increase in the expression of ER alpha divulges the responsiveness of the AKG to the estrogen signaling. So this study provides the first analysis of isoforms of ER alpha during the development of a reptile embryo. This is published in USA. The first paper published in ZZ, which our model has appeared on the cover page of the Journal of Experimental Zoology. First paper, FMF pattern. So then when there is a ER expression is there, then we are interested to know the aromatase, whether there is a aromatase signaling is there or not. So Western blot analysis detected specific band of 58 kDa in the AKG complex, which reveals the presence of AR expression in the embryonic gonad, which is high at FPT than MPT. Means it is high, the aromatase activity is more at the female producing temperature than at the male producing temperature. Then this intensity increases with the increase in the development. So this is the this is the aromatase expression in the medulla. This is ovary and this is the testis, where aromatase expression is more in at the female producing temperature, where it is present in both the cytoplasmic expression in the medullary region as well as in the cortex region, where these are the oocytes, where in case of males it is less, showing only the these are the Leydig cells, where they don't show the aromatase expression, and seminiferous cords a little expression when compared to the females. See, this is the Western blot analysis which reveals the 55 kDa of aromatase in case of females, and it increases with the increase in the age. And apart from that, we got a very interesting result. When we take the embryo section, we always usually take the torso of the embryos, and during this we came to know that the ER expression is present in the neural tube, where neural tube means brain region. It is first expression also analyzed in the neural tube, where here with the increase in the age, the ER expression increases, where it is earlier it is cytoplasmic, then it is nuclear expression, reveals that even brain participates in the sex determination process. And we went for the steroid profile. See, this is the brain testosterone levels in females and in case of males and females. This report shows the sexually dimorphic expression of steroid hormones T and E2 were observed during oviposition, TSP, gonadal differentiation. Very interesting result is we observed that the developing brain of the lizard is capable of de novo steroidogenesis, and it also indicates that it attains potency of synthesizing neurosteroids before the gonadal differentiation, that is during the TSP. Then we are interested whether the brain is also participating acts as an extragonadal source. This study unveils that developing brain senses the temperature and acts as a second site of extragonadal site. First site is we always consider the adrenal. So in conclusion, this lizard exhibit a novel FMF pattern of TSD, which is the first report from our laboratory, and this paper has been published in Journal of Experimental Zoology, Willy Science, and the model has appeared on the cover page. And why this pattern? This PS pattern may be counterbalance the skewed sex ratio, because at different temperatures different sex ratios are produced, and this by exhibiting this temperature pattern, it may be counterbalancing its sex ratio in the nature with alarming light global warming, and the apparent surge in the endogenous estradiol level coincides with the first indication of gonadal sex differentiation. Then we can conclude that estrogen playing a key role. Both testosterone, estradiol are detectable in the developing embryos during TSP. It reveals the synergism between the temperature and the steroids hormone. And immunolocalization of estrogen receptors in the adrenal, kidney, and the gonadal complex during TSP and gonadal difference suggests that estrogen receptor dependent mechanism may be appears to play a key role in determination of the TSD reptiles. Western blot analysis revealed the adrenal, kidney, gonadal expression of two protein bands suggests the occurrence of isoforms of ER alpha. The increase in expression of ER alpha variants during later stages of development diverges the responsiveness of the AKG to the estrogen signaling, which in turn suggests the upregulation of the aromatase, which is playing an important role during the development. High expression level of aromatase associated with the ovarian differentiation further supports our earlier report that can be considered a suitable marker for the ovarian differentiation. So, and recently we got the developing brain of the lizard is capable of de novo steroidogenesis. It also indicates that it attains this potency synthesizing the neurosteroids during the early phases of development prior to the gonadal differentiation. So current research is going, we are after having this, we are going to the gonadal expression of DMRT, MIS, SOX9 in male embryos, and gonadal expression of FOXL2 or SPO, beta-catenin in the female embryos, and to test whether the embryonic brain is capable of this, we got it, and the potential role of neurosteroids in sex differentiation, if any.